Hey guys, how's it going? This is Braxton. We're just going to be talking about homeostasis today. If I could even spell it. Okay, homeostasis. So homeostasis is basically the ability of the body to maintain a relatively stable internal environment, regardless of changes in the external environment. This internal environment that we're referring to is the fluid that surrounds the cells. So let's just say this is cell and this is the fluid. And maintaining this internal environment is crucial for proper cellular function. One example of how it might work in the body is with body temperature regulation. With homeostasis, there's typically a set point. Let's just say right here. This is a set point that the body needs to be at for proper cellular function. That's just when everything's working at its best ability, right? But at times, let's just say with body temp. So body temp was 98.6, that's set point, degrees Fahrenheit, not Celsius, we would be dead. Right there, okay. Like this, okay. And then we have time here. So we have this set point, but obviously external environment, it's hot outside, right? There's gonna be always different changes that are gonna be affecting us on our internal environment. So we won't always be stuck at this set point. So there might be some deviations, kind of in this little bit of a range right here, but it's always gonna be kind of hanging around the set point. Typically, the bigger the range that our body allows us to go from that set point means the less important it really is. But the tighter that range, it's like, oh, this is like super important. We need to keep it at this set point if we're gonna survive. Cool, so our body is able to do homeostasis with a lot of communication. And it communicates through something called a reflex control pathway. Kind of a general kind of structure of a reflex control pathway is first there's a stimulus. Something that changes in our environment. So let's just deal with like body temp regulation, right? Just kind of like just to kind of help it make sense a little bit. So we have our temperature. Either it got really hot or really cold outside. And it's affecting our internal, what the heck was that? Our internal body temp, right? Perfect. So first you have a stimulus, and that's picked up by a receptor. So like on your skin, you're detecting that change, right? These receptors send that signal through an afferent pathway. Either through the blood or through the neurons. I like to think of afferent pathway as arriving with the A to the control center. That's where these receptors send their signal to. Control centers, a lot of people think it's just typically the brain is the control center. It's one of our main ones, but we have quite a few throughout the body. And at the control center, that's where they know what that set point is for different things in our body, like body temp, blood pH, you name it. Those control centers know it. And they'll interpret what really the body wants to do, what we want to do to get back to that set point so we don't deviate too far. So they'll send a signal, again, through the blood or the neurons. It's like the endocrine system or the nervous system, through the efferent pathway. Perfect. So they'll send that signal down to its effectors. These are kind of the guys that are going to get the job done, the effectors. So like your muscles, your organs, whatever you name it, they're going to cause that change in that variable, basically to bring it back to that set point or whatever the body's wanting to do. And then you'll get the receptors picking up on that signal again, and it's just a constant loop. It's always just kind of happening, just to make sure we're staying in homeostasis so we can survive. There are a couple pathways that we use with this idea of a reflex control system. First one, and most common and one of the more important, well, they're both important, but most common is your negative feedback loop. write it right here. Just negative feedback. So with negative feedback, it is basically, it counteracts the change in a variable from the external environment to bring the body back towards its set point. So like, there's a lot of examples of this in the body. There's regulation of the pH in the blood, replenishment of oxygen by the lungs, body temperature regulation. And we'll just stay with this theme of body temperature regulation. So let's just say, okay, right here. It's really hot or cold outside. So the mechanoreceptors, or not the kind of the sensory neurons or thermoreceptors in the skin detect this change in the temperature. So there's that stimulus, which is hot or cold. Okay. Our thermoreceptors pick up on that signal. They send that signal through the neurons or the blood, the sensory neurons, about the temperature through the efferent pathway to the control center. In this case, it'll be the hypothalamus. And it'll be like, okay, we should be at 98.6 degrees Fahrenheit, not percent, Fahrenheit. But we're either getting a little too high from that or we're getting a little too low from that. So let's fix that. So they'll send a signal through the efferent pathway. Efferent pathway to the effectors. And there's quite a few different effectors that you can use. I can't spell that. Through to help with this change in body temp. So it sent the signal through the neurons or the blood. And some of these effectors that can help with body temperature regulation could be like your sweat glands. So let's just say sweat glands, which will start to sweat. It'll receive the signal from the hypothalamus, begin to sweat, and the sweat evaporates and cools the body down. It transfers that heat to that sweat, which will leave our body. We also have our blood vessels, which can do quite a couple different things. So if it's too hot, let's just say it's too hot, then they will vasodilate, which is a fancy word that I just learned. That means that the veins or your blood vessels will dilate or get bigger, basically, to allow more blood flow to go to the skin, and this will allow that heat to be transferred to the external environment. If it's more cold, then they will vasoconstrict, which will pretty much just make it so that, the veins, blood vessels, arteries, right, they will constrict and keep that blood from going to the surface, like around the skin, so that heat can be kept within the body if it's getting really cold out, right? We don't want to lose that heat. There's also signals that the skeletal muscles can receive. And these signals will cause them to shiver. It's causing them to contract, and it's like, you're shaking, right? Your muscles are contracting, and it's heating your body up. So that's basically what an example would be for negative feedback. If you're getting too hot, they'll do the opposite and try to cool you down. If you're getting too cold, from the set point 98.6 degrees Fahrenheit, it'll do the opposite of whatever's happening and increase the temp to get back to that set point. Perfect. We also have positive feedback. So when negative feedback isn't working adequately, positive feedback will jump in. Typically means that something's broken, something's not working, it's not good. So typically, positive feedback is unintentional. But about 5% of the time, very low amount of the time, it is actually intentional. We want to do that. And a couple of these cases are with childbirth and blood clotting. So for positive feedback, it's basically the response to the stimulus that we're doing is increasing the deviation from the set point. So instead of negative feedback where you're leaving that set point but you're going back to it, you're trying to always return to it, Positive feedback would be just continuing to leave, further deviations from that set point. Okay. Let's talk about childbirth. This is a good example of positive feedback. So I'm going to draw a little picture here. So we have a little baby, or sometimes a big baby, that will be in the uterus. So this circle right here is the uterus. I'm going to write baby. Perfect. And then we have the cervix, like right there. So what's happening is that as the baby's starting to grow, they'll begin to push out on the uterus. And there'll be these stretch receptors in the uterus and the cervix that will pick up on the stretch, especially in the cervix here. This will send a signal through the afferent pathway to the control center, which would be the brain. And it's like, holy cow, this baby's growing. We're stretching. What do we do? Well, the brain's like, oh, that's good. We want to get rid of that baby, like have it be born. So we're going to release through the efferent pathway, or the exiting pathway, right? That's how I like to think of it. We're going to release oxytocin. And oxytocin is known to increase the contractions, which will create further stretching and pressure and stimulus on that cervix. So this will just keep exciting that whole thing. It'll just keep going back and forth till that baby's out. So this is basically what happens with positive feedback. It sends that signal through the afferent pathway to the brain, and because there's stretching on the cervix, so it'll go through the blood and neurons. And then the brain, or the control center in this case, will release oxytocin through the efferent pathway, through the bloodstream, and that will go back to the uterus to increase the contractions. So today, just a little recap. We talked about the definition of homeostasis and how it might work in the body. We also talked about the reflex control system, right here, the components of it in each one. And then we also talked about negative feedback, as well as a couple of examples, such as regulation of pH in blood, thermoregulation in the body, and then we talked about positive feedback as well. Perfect. This is just part A of essay exam 1. Okay, right now we're going to talk about the 3 types of protein-mediated transport that we discussed in the class this week, being diffusion through channels, carrier-mediated diffusion, and active transport. To start, just to kind of give an idea right here. So here's like a giant container I have, and I'm separating that container with a little thin semi-permeable membrane. We're going to call this side A, and we're going to call this side B. So on one side, I add a lot of whatever this is, just a few kind of like 4 different particles, right? Because everything has kinetic energy, everything's moving, slightly, and everything's going to want to reach a point of equilibrium where everything's pretty much the same on each side. So with time, this semi-permeable membrane that allows the movement of these 4 particles will eventually allow equal concentrations of both of the particles to be on both sides, from the A to the B. Okay, so now we're going to talk about diffusion through channels. Diffusion through channels is one way that we're able to move these particles, or we can call them solutes, either in or outside or leaving the cell. So here's a little cell right here. I'm going to draw a few of the different types of channels that we see within our cell membranes. So we have right here just a channel protein. So this is just a normal channel. Let's just say it's just a potassium channel, right? With these channels, they're very important for allowing specific solutes to move down their concentration gradient from high to low. So for example, for potassium right here, there's a ton of potassium within the cell. And there's not very much outside, so I'm going to draw a little tiny potassium. it's going to want to reach a point of equilibrium, which is where there's equal concentrations of potassium inside and outside the cell. So this channel protein allows movement of this potassium to go outside the cell. With these channel proteins, they can be for specific ions. So in this case, it was with potassium. Other ones only allow sodium, which is Na+. Others will only allow chloride, and so on. There are other types of these protein channels as well. There are ones that are have little gates on them. So you have your little one right here, and some of them just have a gate to prevent things from going from their high concentration to their low concentration. There are ones that are voltage gated, so changes in voltages will allow that gate to open. There's also, I'm just gonna draw them like this actually. There's specific signal molecules or gates where you need like a specific, I'm just gonna draw like something like that, where something needs to bind to this gate for it to open. And then there's other ones that are called mechanically gated channels, where there's like a stimulus such as like stretching, compression on the membrane, whatever it is. And I'm just gonna go like, I'm gonna draw something like that too. Okay. But these types of channels, so the diffusion through channels, they are a passive process. What they use is the concentration gradients from high to low to move the solutes. And when I say passive process, it means that ATP is not required. Kinetic energy alone will allow the particles or the solutes to move from a concentration of high to low to reach equilibrium. Things that can affect the rate pretty much of these channels, this diffusion through channels, could be the concentration gradient of the solute. So that say there's just like so much over on this side and so little over here, it's gonna be way more enticed to move over to that side, as if they were already, instead of if they were just already pretty close in concentrations. And then the number of channels. So if I have more channels that could allow, for example, potassium to leave the cell, it's gonna happen a lot faster. Sweet. I'm just gonna move down. Okay. Now we're gonna talk about carrier mediated diffusion. Sorry, I'm trying to write this quick. Okay. So with carrier-mediated diffusion, carrier proteins bind to specific solutes on one side of the membrane, and the binding causes a change in the shape of the protein, which moves the whole protein, carrying that solute to the other side of the membrane. So it's kind of pretty much the same thing. So you have your cell right here, and I'm trying to move in, I don't know, let's just say sodium, for example. I have a little protein right here, and it's got a little spot for sodium to bind onto. Sodium won't be able to get through this membrane because it's a phospholipid bilayer. It doesn't let many things really get through unless through it like a channel or through proteins getting onto the other side. So this protein will bind to the sodium and move and cross over with the sodium to the cell, or just whatever solute really you're talking about. And it could be moving from the cell to the outside of the cell, or from the outside of the cell to the inside of the cell. So I'll just say solute right here. Things that can affect pretty much the rate of these as well is the number of carrier proteins. So reaches a point where, one second. Okay, so the rate of, of your diffusion here in carrier-mediated diffusion, or called facilitated diffusion, it'll go up as the concentration. So like as I add more of my solute, it's still going to be going through the high to low diffusion there. But since we're depending on these carrier proteins to allow the solute to cross that membrane, it's going to really depend on how many carrier proteins we have. So it's going to go up at a constant rate, but once we're using up all those carrier proteins, it's like, crap, we don't have any more to give. Like, we used our whole team, we can only go so fast. So it kind of reaches a point where it just levels off, where it can go only so fast. Even though we have like say we're in an assembly line, handing everything, carrying these things, like all this food to another side of the whatever building you're in. And then even though there's a ton of food, we keep adding a lot more food. Trucks are coming with food, but we only have like 10 workers. We can only go as fast as the 10 workers can move, really. So that's how I kind of think of it. Perfect. And then, so yeah, with carrier-mediated diffusion, it still doesn't use energy. It still relies on a concentration gradient. So no ATP here is needed, really. Okay, and one thing that can also affect the rate is competition. So if there's a lot of different, if there's several different solutes, so say solute A and solute B, that can bind to the site right here, they're going to compete for that. So let's just say that solute A is a little bit better at binding at this spot. Its rate is going to be faster than solute B because it's just not as good as solute A is binding to this carrier protein. So if they're both competing, solute A is going to win. It's going to be a lot harder for solute B to go faster. Okay. Now we're going to talk about active transport. So there are two types of active transport. First, we're going to talk about primary active transport. Okay. The main thing with primary active transport is that it does require energy because it is moving the solutes against their concentration gradient. I'm just going to do this for concentration. Their concentration gradient. One example of this is a really important one, actually. It's our sodium-potassium pump. So I'm going to draw it right here. So inside the cell, we have a lot of potassium. And then on the outside of the cell, there's generally a lot more sodium. Perfect. So we have our pump right here. Let's just draw it right here. So with our sodium-potassium pump, it puts out three sodium ions and then brings in 2 potassium ions. And so for these, there's already a lot of potassium in here. Normally, since there's a lot more potassium inside the cell than there is outside the cell, you would expect that potassium would want to leave, right? But with the full help of ATP, we get help from the ATP to be able to push against this concentration gradient and push the potassium into the cell. And it's the same thing with sodium. Normally you would want sodium to go in with that concentration gradient, but we're pumping against that, and that ATP is going to provide that energy for that. Awesome. Yeah, and it'd be the same thing for specificity. So there's certain types, for all these, there's certain types of proteins that will bind to different things. So there's, or even the channels, only certain things can go through those, such as like up here. Like you have like a potassium leak channel. Only potassium can go through that. You have a sodium one as well. Only sodium can go through that. So things are very specific and in order. There's also something called a secondary active transport. Active transport. So this is also going against against a concentration gradient. That's a long word. Okay, I'm just gonna stop right there. So against a concentration gradient, it's kind of like you're trying to push up a hill, right? It's a lot easier if you're just trying to run down the hill. You can roll down and it takes no energy. But if you're trying to go up the hill, it's just like, oh my gosh, that's like so tiring. It takes a lot of energy. It's the same thing here. You need some source of energy. The thing with secondary active transport that separates it from primary is that it's not directly using ATP as an energy source. What it is using is energy stored in the concentration gradients to move the desired solute that we're trying to move. So before we were talking about this, the sodium potassium pump right here. And what this is doing is it's creating that even bigger concentration gradient. It's creating even more sodium on the outside than the inside of the cell, and the same thing with potassium on the inside of the cell than there is on the outside. So this is creating even more of a drive for those sodium to want to go inside the cell, right? So, which was created by the use of ATP originally. So since sodium is in higher concentration outside the cell due to the primary active transport, the sodium gradient is typically used to power the secondary active transport. Carrier proteins, so they'll also still use carrier proteins right here. Let's just say, bloop, 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 right there. That's at the attachment site. So they'll have an attachment site for both sodium right here, and then for whatever other solute we want to use. Cool. So what the secondary active transport does is that this will bring the sodium into the cell, allow it to use that concentration gradient where it's like, there's so many of us out here, we want to get in there. So it'll just make its way inside the cell, and it'll bring the solute with it. This would be called a symport. So if something's going in at the same time with another solute, like the sodium, it'll be a symport. But this also can do the same, where sodium will use that concentration to go into the cell, and it'll drive out whatever solute we want, a specific solute. And that would be an antiporter or an antiport. But yeah, the main thing with this one is that ATP is needed to generate the sodium concentration gradient, but it's not directly involved in moving the desired solute across the membrane. And that's pretty much it with this part 2 with protein-mediated transport. Okay, for this final part of this essay, I'm going to talk about more of osmosis. Osmosis is very similar to diffusion, except it's purely just dealing with water. Osmosis is the movement of water across a selectively permeable membrane from an area of high concentration to an area of low concentration. So basically the water is moving from the side with lower solute concentration to the side with higher solute concentration until it reaches equal concentrations or equilibrium. So I drew out this picture. I'm not sure what this thing would be called, but it's kind of just a little container. Right here I'm putting a semi-permeable membrane that's only permeable, or in other words, only lets water cross through it, but no solute. So on both sides, I'm going to put equal amounts of water. And then on this side, I'm just going to throw in some solute. Let's just say I put 4. But on this side, I decided to maybe just double it. I'll just throw in a lot more. Oh, the idea is that this side has more solute than this side. Okay. So with osmosis is going to want to basically equalize those ratios of solute to water. So since this side has a lot more solute in comparison to water than this side does, the water is going to want to move from this side over to that side, because they're like, holy cow, there's so much solute here, we're outnumbered. Well, it's like, okay, we'll send help, we have extra people. Goes through that permeable membrane, and what it does is creates this difference in the amount of water in there until it reaches a state of equilibrium. So it reaches a state where pretty much the ratio between the water and this solute is equal to this side. And it creates a change there. So pretty interesting. Cool. So that's osmosis for you. Just a quick little lesson there. Now we're going to talk about osmolarity. So osmolarity, so there's different ways we can measure concentrations in different solutions. So let's actually talk about molarity first. So this one's should be familiar to you if you've taken chemistry. So it's molarity, we represent it with capital M. Moles of solute. So moles is just basically a number that represents molecules per just a number of molecules. It's 6.02 times 10 to the 23 molecules. It's just a huge number, so we just reference that to just 1 mole, just to simplify things. So like this could be, for example, let's just use NaCl for these. So NaCl, this is 1 molecule of NaCl, or let's just say this is 1 mole of NaCl. This is going to be our solute. We measure that in liters of solution. And our solution is our solvent, which would be our water in this case, plus the NaCl. So just like the whole cup. If we had a cup, includes the water with the NaCl in there. Cool. So this is just one way we measure concentration. What it doesn't account for is ions or dissociation of ions. So when sodium or NaCl is placed into water, it can actually separate nicely into 2 different things, into a sodium ion and a chloride ion. So now there's pretty much like the double amount of things inside the solution. It's not just like one thing, like NaCl, it stays together, it's happy like that. It's going to separate. So now there's just more influence of different things within the solution. We would call this in chemistry at least like a von Hoff factor, which accounts for this dissociation of ions. More complex ions like glucose, this doesn't dissociate very nicely. So it just stays as this. So that would have like a von Hoff factor of 1. 1 ion that's really making a difference in the whole thing, 1 particle. But this one would have a von Hoff factor of 2 because it's dissociating into 2 different particles or ions. So it's going to have pretty much double the effect on things. So to account for this happening, this dissociation, we have osmolarity, which I believe is just os like that. And that equals moles, or sorry, that's number of particles per liter of solution. So hopefully that's clear what the difference there between molarity and osmolarity. Okay. Now we're going to talk about osmolarity versus tonicity. So when we're talking about osmolarity, we're pretty much describing the total solute concentration of a solution compared to that of a cell. So a lot of the time we're comparing things between the cell and a solution. So each thing has their certain, concentration of solutes or different things, different osmolarities. Normally, the cell has an osmolarity of 300 milliosmoles inside of it, right? This is just the number of concentration or number of particles it has per liter. So that's its concentration there. But solutions can have different types of concentrations. It could have more particles in it, or it can have less. When our concentration is equal, when it has the same number of particles in the solution as it does compared to the cell, we call this isosmotic there with an S right there. When the concentrations are equal, I'm just going to do equal. This is concentration right here. Okay. Now, when it is, when the solution, so it's really just a point of comparison, really. When the solution has more particles per liter or a higher concentration or higher osmolarity than the cell does. So let's just say it had 400 milliosmoles. Okay. Then this would have a higher concentration than the cell. So to kind of describe that reference, we call it hyperosmotic. Hyper just means more. Perfect. And then to describe when the concentration is less than the cell, so we have 200 here and then 300 here, comparing the solution to the cell, we would call the solution hypoosmotic to the cell. This is a hypoosmotic relationship. Okay, perfect. So that's describing more osmolarity. It's just describing the total solute concentration of a solution and comparing that to a cell. Now we have tonicity. Tonicity gets a little bit more tricky, but it's not too bad. Tonicity describes more so how a solution affects the volume or shape of the cell due to the concentrations there. So now with tonicity, so with osmolarity, we're kind of just looking at, we just care about the concentrations of both the cell and the solution before anything even happens. But as we talked about earlier with osmosis, this changes things. It's going to cause movement of water from the solution to the cell and vice versa. So with tonicity, we can have different types of kind of terms for tonicity. We can have isotonic. This is where, so we're going to go like this. We're going to have a little bit bigger of a container. Like that. So we have our cell. That's 300 milliosmoles. Okay. So in this isotonic solution, it has the same concentration of non-permeable solutes as the cell. So non-permeable, I mean, is just like the solute's not going to make its way into the cell pretty much. So this one has 300 milliosmoles, and then this one the solution has 300 milliosmoles. Like that. So it'd also be isoosmotic, just because they have the same number of concentrations. But since there's the same number of concentration, the water's happy. It's like, no, there's no reason for me to move over there, no reason for it to leave the cell. Nothing's going to happen to the cell. It's just going to stay the same shape and size. Now let's say that this one the solution has 400 milliosmoles. So more particles per liter of solution than the cell. This would cause the water to be like, holy cow, they're getting outnumbered in the solution. We got to go help them. So the water will move outside the cell into the solution. And this would make it a hypertonic solution. And the effect that this would have, and hyper just means pretty much a higher concentration outside the cell than the inside. But this is more talking about the effect. Water will leave the cell and go into the solution, so it'll cause it from going like a nice pretty cell like that to being kind of like, it's like all dehydrated and gross. So that will happen in a hypertonic solution. Now we have, let's pretend here. Okay, so now we have our third scenario. So now we have it where, let's say, so the cell has 300 milliosmoles. Let's just say the solution had 150 milliosmoles. So kind of review from osmolarity, this would be a hypoosmotic solution because it has less solutes than it does in the cell when you're comparing the two. So since water's like, sorry, that was the same example. I think I went backwards right there. Let me just maybe cut that out. Okay. Anyways, okay. So this is it's a 150. So let's just say we have less concentration inside in the solution than the cell. So since it has a less of a concentration, oh no, it's right. Okay, I'm right. Okay, so this has less of a concentration, this one has more. So it's like, crap, the water in there is getting outnumbered. We have so much extra water per thing. So now we can go into the cell with the water. This will cause a swelling of the cell. It'll go like, and it'll even sometimes burst. I don't know how to draw that, but that'll happen there. So this would be a hypotonic solution. So there's less solute as compared to that inside the cell. So the water will move into the cell. Okay. Hopefully that makes sense there. Now it gets kind of weird with sometimes you can get it where a solution can be isoosmotic. Let's see if I spelled that right. Osmotic. But it could be hypotonic. And you should be thinking like, what the heck? That doesn't make sense. If it's the same concentration, why would the water want to move? Because they're all happy, right? Well, there's some solutions that are, or solutes that are permeable. They're able to cross that membrane. And one example is dextrose. So we have 5% dextrose is our example that we're going to use. This is just pretty much glucose, a glucose solution. And if you remember from bio, glucose is metabolized by the cell to create ATP. So it uses glucose as a source of energy. And 5% dextrose is equal to 300 milliosmoles of particles. So when you have this in here, you're like, okay, like that looks the same. Why would, so there, this is isoosmotic, but why would it be hypotonic with the solution compared to the cell? The reason for that is that with dextrose, since the glucose is able to cross that membrane, it's permeable to that membrane, it will make it in there. So it'll go into the cell here, and then it's going to be metabolized by that cell. So it's going to keep using that, and it's going to create a concentration gradient with that. Because if it's going into the cell and it's being used up, then there's going to be less of the glucose inside the cell than there is in the solution. So I'll just keep driving that to keep going because it's going to keep getting used up. So I'll keep pulling it in. But then we'll run into a problem that it's pretty much like all this was used up. So now we're at like 0 osmoles, milliosmoles in the solution, and then we're at 300 milliosmoles of non-permeable solutes. So now it's like, holy cow, we are so overpowered with solute in this side inside the cell. We need help from water. And the solution is like, we don't have any solutes in here, so let's go send all our water. So they'll follow the glucose. And it'll cause the cell to swell and possibly lyse. So this is just an explanation of osmosis, osmolarity versus tonicity, the effects on the cell with different types of, like when you're talking about tonicity, and how a solution could be isoosmotic, but also hypotonic. And that will be it for this exam essay.